Hello, everyone. This is Dr. Kuo, and today we'll begin our lecture series on urinary system. So this is the list of topics that will be covered in this lecture. Now, briefly about the function of the kidney. Our kidneys are involved with regulation of the extracellular fluid, including blood. And so it's also involved with the regulation of blood pressure. Furthermore, kidneys are involved in regulation of blood osmolarity with maintenance of electrolyte balance, with the regulation of acid-base balance in our body, so pH, as well as excretion of metabolic wastes, and finally, producing hormones. Now, this is a quick anatomy review of the urinary system. Our urinary system consists of the two kidneys, ureter, bladder, as well as urethra. The kidneys are located retroperitoneally. Okay, so behind that parietal peritoneum, you have two, one on each side. They are bean-shaped, and each of them consists of two regions, the renal cortex and the renal medulla. Nephrons are the functional units of kidney, and it consists of renal corpuscle and renal tubules. The renal corpuscle includes the glomerulus and the glomerular capsule, whereas the renal tubules consist of proximal convoluted tubule, the loop of Henle or nephron loop, including the descending limb and ascending limb, okay, and the distal convoluted tubule. Okay. And you can see here that the distal convoluted tubule of different nephrons merge into the collecting duct. Okay, and also make note of which region, whether it's in the renal cortex or renal medulla, each of these structures lie in. Okay, so in other words, notice that renal corpuscle, proximal convoluted tubule, distal convoluted tubule, they're all located within the renal cortex, whereas in the renal medulla, you will find the loop of Henle. Also note that the collecting duct spans through the cortex as well as the medulla, so renal cortex and medulla. And there are two primary types of nephrons. There are the cortical nephrons with the shorter loop of Henle. We also have the juxtamedullary nephron, which have these really long loop of Henle. Okay, and over 90% of the nephrons in our kidneys are these kind, the cortical nephrons. Okay, but the juxtamedullary nephrons do also play a very important role in modulating the osmolarity of our urine. Okay. As you can see in this image, each nephron contains or is associated with two arterioles, as well as two sets of capillaries that form a portal system. So recall that a portal system is consists of two capillary beds lying in series. Okay, and in this renal portal system, we can see that the first capillary bed is that glomerulus. Okay, and the second capillary bed is that peritubular capillaries. And in the case of juxtamedullary nephron, we have our modified peritubular capillaries called the vasa recta. So vasa recta is essentially a type of peritubular capillaries. Okay, but they are specific to the loop of Henle for these juxtamedullary nephrons. Okay, so overview of kidney function. There are a few different separate renal processes involved in the formation of urine. We have glomerular filtration happening across the filtration membrane within the renal corpuscle. We also have tubular reabsorption and tubular secretion happening throughout the renal tubule. Okay. And then after all these happen, the fourth process is excretion, which involves getting rid of substances from the lumen to outside the body. Going back to this image, you see that filtration is a process that only happens within the Bowman's capsule, so within the renal corpuscle, across this filtration membrane. Once the fluid has entered into the tubular lumen, it can either be reabsorbed or it can continue and then end up in the collecting duct. Okay, so if it's reabsorbed, then it won't be excreted later on. But if it's not reabsorbed, it stays within the lumen and will later be excreted from the body. Okay, and besides from reabsorbing from the lumen into the blood, we can also have secretion, which takes substances in blood and then puts it in the tubular lumen. Okay. It's important to note that when the fluid enters into the tubular lumen, at this point we call it filtrate. We don't call it urine. Okay, and this is because that filtrate composition changes significantly by the end of the distal convoluted tubule. Okay, so the composition of our urine differs drastically from the filtrate that we see at the start of proximal convoluted tubule. So that fluid within the glomerulus generates hydrostatic pressure that promotes the process of filtration within the Bowman's capsule. Only 20% or 1/5 of plasma that enters into glomerulus ends up being filtered. Okay, and that's just the amount that gets filtered. Okay. And we'll see at the end of our collecting tubule or collecting duct, only less than 1% of fluid ends up being excreted. Okay, so of all the fluid that gets filtered, only 1% of that ends up being excreted. Okay, and this is because over 99% of it gets reabsorbed throughout the renal tubule. We're now going to take a closer look at the process of filtration. Recall that filtration membrane consists of that simple squamous epithelium from the glomerulus, as well as the foot processes of podocytes lining the glomerular capsule. Okay, and also sandwiched in between the two layers is a thin layer of basement membrane. Okay, so that means for a substance to enter into Bowman's capsule, it has to first cross through the capillary epithelium, the basement membrane, and then the filtration slits created by the podocytes. Okay. If we take the plasma volume entering into afferent arteriole to be 100%, we'll see that only 20% of that 100% enters into Bowman's capsule. Okay, in other words, it's filtered. Okay, and this of this 20%, much of it gets reabsorbed and gets returned back to the systemic circulation. Okay, whereas only less than 1% of the volume is excreted into the external environment. So what that means is over 99% of the plasma that enters into the kidney ends up being returned to the systemic circulation. Okay, so your kidneys are really, really good at retaining volume. Okay, otherwise you would quickly dehydrate to death. There are two types of forces that matter in glomerular filtration: hydrostatic pressure and oncotic or colloid osmotic pressure. Hydrostatic pressure within glomerular capillaries is going to act to promote the process of filtration, whereas hydrostatic pressure within the capsular space is going to act in the opposite direction and making it harder for filtration to occur. Okay. Oncotic pressure within the capsular space is also going to promote filtration because they act as little sponges to promote fluid entering into the capsular space. Oncotic pressure within the glomerular capillary, on the other hand, is going to help retain water within the glomerulus. In other words, prevent filtration. So if we balance all of these factors, okay, and plug in the usual values or the approximate usual values for each. And by the way, oncotic pressure in the capsular space is usually around 0 because proteins are generally not filtered. Most proteins are not filtered, with the exception of some, but most larger particles, larger proteins, as well as cells are not going to be filtered, okay? And therefore, oncotic pressure within the capsular space is around 0. So if we balance out all 4 forces, we'll see that the net filtration pressure is around 10, okay? And because this value is above 0, that means there is a net filtration happening as opposed to net reabsorption. And one key feature to note about the afferent and efferent arteriole is that you'll see that the afferent arteriole is wider than the efferent arteriole. Okay, and this creates a positive and high hydrostatic pressure within that glomerulus that contributes to filtration. The volume of fluid that filters into Bowman's capsule per unit time is known as glomerular filtration rate. Average GFR values is around 125 milliliters per minute, or around 180 liters per day. That means since you only have about 3 liters of plasma in your body, that means the kidneys are able to filter your entire plasma volume around 60 times a day. That's an incredible amount. Okay. And if it wasn't because of the fact that most of that filtered volume ends up being reabsorbed, then we would become dehydrated and die of dehydration within half an hour. So GFR is influenced by 2 factors: the net filtration pressure, NFP, and the filtration coefficient. Now, the net filtration pressure we've already discussed is basically a balance between hydrostatic pressure and the colloid osmotic or oncotic pressure in the glomerulus versus these forces within the Bowman's capsule. Okay, and net filtration pressure is determined primarily by renal blood flow and blood pressure. Okay, think about what that means. Blood pressure, as your blood pressure rises, that also generally mean the rate at which blood is entering into your kidneys is going to increase, and that's your renal blood flow. Okay, and so higher blood pressure, okay, it's going to lead to higher net filtration pressure because of the hydrostatic pressure. Okay, and now the filtration coefficient has 2 components: the surface area of the glomerular capillaries that's available for filtration, and the permeability of interface between the capillary and the Bowman's capsule. Since blood pressure provides the hydrostatic pressure that drives glomerular filtration, you may expect that as blood pressure rises, glomerular filtration rate, or GFR, would also increase. And if blood pressure falls, you may expect that GFR would decrease as well, but that is actually not the case. Instead, GFR is actually maintained at a really tight range, and it's able to stay remarkably constant over a wide range of blood pressures. And as long as your mean arterial blood pressure stays between 80 and 180, GFR generally stays the same at around 180 liters per day. And the control of GFR is primarily done by regulating the blood flow through renal arterioles. That means if we change the resistance of the renal arterioles, okay, whether it's the afferent arteriole or efferent arteriole, we can change the renal blood flow, how much blood goes into the kidneys, okay? And if we increase the resistance of renal arteriole, less blood will go into kidneys and more blood will be diverted to other organs, okay? As for the effect of changing resistance of arterioles on GFR, that's something we'll discuss on the next slide. Okay. The effect of increased arterial resistance on GFR depends on exactly where the resistance is being changed. If the resistance is increased within the afferent arteriole, hydrostatic pressure will decrease on the glomerular side of the constriction. So this would lead to a drop in GFR. If resistance is increased in the efferent arteriole instead, the hydrostatic pressure in the glomerular capillaries would actually increase. Okay. And increased glomerular pressure leads to increased GFR. So you can see that depending on whether we're increasing resistance within afferent arteriole versus an efferent arteriole, the results are actually the opposite. Here, right, we are actually decreasing GFR. Here, we end up increasing GFR. So how is our body able to regulate glomerular filtration rate? Well, there are two categories of mechanisms in place. We have our intrinsic mechanisms and then extrinsic mechanisms. The intrinsic or intrarenal mechanisms involve structures within the kidney, okay, whereas extrinsic mechanisms involve structures outside the kidney. And when your mean arterial pressure is between 80 to 180, the intrinsic mechanisms are the predominant mechanisms, maintaining your GFR, okay? Whereas when, say, you are in a car accident and bleeding a lot, okay, and your mean arterial pressure falls really low, this is when the extrinsic mechanism will kick in place and to try to maintain your blood volume and blood pressure such that your other tissues can continue to get blood supply. Okay. And so the mechanisms that fall under the intrinsic category are the myogenic response and tubular glomerular feedback. And so we'll talk about these two mechanisms first, and then in the next lecture, we'll go more into the extrinsic mechanisms. This is another summary diagram going over that information we just talked about, but in more detail. So intrinsic mechanisms, we have myogenic response and tubular glomerular feedback, whereas extrinsic mechanism involve either neural controls through the sympathetic nervous system or through hormonal mechanism called the renin, angiotensin, aldosterone system, which we'll talk about next time. Okay, so first we're going to look at the myogenic response. As we mentioned, the myogenic response is the way our kidney is able to regulate its own GFR. And it relies on the intrinsic ability of the vascular smooth muscles to respond to pressure changes. What that means is when the afferent arteriole gets stretched and the smooth muscle in its wall gets stretched, these smooth muscle cells respond by contracting and therefore constricting the afferent arteriole. Okay, so it's just as simple as that. When the smooth muscle cells within afferent arteriole are stretched, okay, and that happens when you have increased renal blood flow. Okay, and these smooth muscles then will respond by contracting and thereby causing vasoconstriction of the afferent arteriole. And so what these do is at high blood pressure, this autoregulation method protects your glomerulus from being damaged by high blood pressure. Okay, and when your blood pressure is low, it will respond by dilating, and therefore ensuring that there will be sufficient blood flow entering into the glomerulus, and therefore your waste will still be removed from the body. Now, moving on to tubular glomerular feedback. Tubular glomerular feedback is a local control mechanism where the fluid flow through the renal tubule influences the GFR. Recall from renal anatomy that the structure of the juxtaglomerular apparatus, JGA, created by that final segment of thick ascending limb loop of Henle, passing in between the afferent and efferent arterioles, and where the tubules and arteriolar walls are modified in these regions where they contact one another. So each JGA consists of these specialized cells. We have macula densa cells. These are modified tubule epithelial cells of the thick ascending limb loop of Henle, which makes contact with the arteriole of the glomerulus from which it arose. And these cells, they are capable of detecting the sodium and chloride that pass through the lumen in this filtrate. We also have granular cells over here. Sorry for this is displaced. We also have granular cells over here. Granular cells, also called juxtaglomerular cells or JG cells. These are located in the afferent arteriole of JGA, and they are specialized smooth muscle cells that are capable of secretion of renin, so secreting renin. Okay, and renin, as we know, is an enzyme that's important for salt and water balance in our body. Now, when the sodium and chloride delivery past the macula densa increases as a result of increased GFR, macula densa cells will send a paracrine message to the neighboring afferent arteriole. Okay, and the afferent arteriole then responds by constricting, thereby increasing the resistance and leading to a decrease in GFR. So we've just covered these two methods that fall under the intrinsic category of mechanisms for GFR regulation. And the extrinsic mechanisms is something we'll talk more about in the next lecture. But for now, we're going to move on to talking about reabsorption. Moving on to talking about tubular reabsorption. Tubular reabsorption is the movement of tubular contents, whether that's water or solute, back into blood. And 99% of glomerular filtrate, meaning the solutes that get filtered through the filtration membrane and ends up in the proximal convoluted tubule, 99% of that, okay, is reabsorbed by the nephron back and put back into the capillary. Okay, and water then later follows by osmosis. Okay. And if it wasn't because of that, our body would actually dehydrate within less than half an hour. Okay. In other words, if your body wasn't so good at reabsorbing all these solutes, and then with water following by osmosis, and then your body would lose all its fluid volume within less than half an hour. Now, these tubular cells have polarity to them. We've got the apical side, and then we have the basolateral side. And tubular reabsorption can either occur through a transcellular route, meaning it passes through the apical membrane and then the basolateral membrane before it reaches into the circulation, or it can utilize a paracellular route in which it's passed through in between the cells. Okay, so the steps of tubular reabsorption. Well, sodium being the most abundant cation in filtrate, its reabsorption is through a primary active transport mechanism. And in other words, it's using this sodium potassium ATPase to pump sodium against its concentration gradient from a place of lower concentration to a place of higher concentration. And then the sodium then diffuses into the circulatory system, whereas potassium is pumped into the cell, maintaining a high intracellular potassium concentration. So this pump uses ATP directly. So this is a form of primary active transport. Now, because of this pump, we're able to maintain a low intracellular sodium level inside the cell. And this actually allows for the secondary active transport of other solutes. So secondary active transport is a transporter protein that couples the movement of an ion, usually sodium or hydrogen, down its electrochemical gradient to the uphill movement of another molecule or ion against concentration or electrochemical gradient. And this type of transport is indirectly dependent on the sodium potassium ATPase activity. So this is what secondary active transport is. Okay, so in other words, we're using the potential energy stored in the form of sodium gradient to drive the uphill movement of these other solutes. And this is how glucose is reabsorbed. This is how amino acids, as well as some ions and vitamins, are reabsorbed. Okay, and then following the reabsorption of these solutes, we have the passive water movement through osmosis. So this is a form of passive transport. It does not require energy. Now, once water is reabsorbed, it increases the relative concentration of the other solutes that are left behind within the tubular lumen. And these other solutes can then be reabsorbed as they move down their concentration gradient through passive transport. And that includes lipid-soluble substances that can be transported through transcellular route, so directly by diffusing across the phospholipid bilayer. And then the other ions, like calcium, potassium, chloride, as well as urea, can then diffuse through the paracellular route back into the circulation. Since our tubular cells have finite number of carrier proteins on their membranes, that means transcellular transport systems are limited. Okay, and so transport maximum, TM, this is the point at which increase in tubular concentrations of a substance will not result in an increase in its reabsorption. Okay, and this is because when all the carriers or a specific solute are saturated, that means not all the solute can then be reabsorbed, and whatever wasn't reabsorbed will end up being excreted in urine. And this explains why patients with severe hyperglycemia, perhaps undiagnosed diabetes mellitus, can be found to have glycosuria or glucose in their urine. Okay, even though in all of us, the glucose is freely filtered into the Bowman's capsule and enters the proximal convoluted tubule, but because our kidneys are typically really highly efficient at reabsorbing all the glucose, 99% of it is generally reabsorbed by the end of proximal convoluted tubule, okay, such that we don't see any glucose in our urine. However, when these patients untreated diabetes with or undiagnosed diabetes, when they have such high blood glucose, okay, all those glucose overwhelms the carrier in the tubular lumen. Okay, and whatever glucose couldn't be reabsorbed in time ends up being excreted in the urine, and then water will also follow by osmosis, which means these patients will typically be pretty dehydrated as well. And that concludes today's lecture. We'll see you in urinary system part 2.